Hello?
To make you comfortable. Or I can just share my phone. You can put it on the table. Yeah, this is the I don't want anybody to trip back here. Thank you.
Hello, everyone. We're going to be getting started soon.
Right here, Alex. The audio. I think it's because we're down to one speaker. One speaker. This many people.
Now I can unmute. Can you see me? Can you
For
hear
those
me?
on Zoom, we'll be getting started shortly. For those
Perfect.
in person, if you could
I
please.
think I'm gonna. Should we start?
No, they're just. Don't be shy. Okay, hey. Wait, you acronym for what? Like acronyms, like things like yeah, exactly. You're leaving me that top in the will, right? That is lovely. Saying, make sure if he says something you think people don't understand or any clarification, stop it, interrupt it. Absolutely. I'll be on it. Well, I think we're good. Okay, well, yeah, the kid, yeah, this is like background science for my questions. So, yep, I'm good.
All right, he's just getting going.
Oh, so should we wait? Okay. We're going to start in another minute. 1 minute, 60 seconds. Everyone hear me? I'm going to ask everyone to quiet down. We're going to settle down and get started. Ooh. How's this mic sounding? Is it okay? Okay. Good evening, everyone. Good
مساء
evening.
الخير جميعا.
Can I ask my team in the back to have people in the back quiet? We have to quiet down. Thank you. Have to do like an elementary school. Shh. See, look at that. It always works. Good evening, everybody. Thank you so
مساء
much
الخير
for being
جميعا.
here. Let's
شكرا
get
جزيلا
started.
لحضوركم
A round of applause just to get us
وتصفيق
up.
حار
Thank you
للجميع.
so much.
شكرا
My name
جزيلا.
is
اسمي
Dr. Haley Thompson.
دكتورة هيلي تومسون.
Oh, hold on.
لحظة واحدة.
We are having this translation
نحن
in
نقوم
Spanish and Arabic. So
بالترجمة في اللغة الإسبانية والعربية.
we're good? Okay. See, my team is on it. So good evening, everybody. Can everybody still hear me? I'm Dr. Haley Thompson. Thank you so much for being here this evening. I have the honor to serve as Associate Center Director for Community Outreach and Engagement at Karmanos Cancer Institute here at Wayne State University. Just to share a little bit about Karmanos Cancer Institute, we are what is called an NCI, National Cancer Institute, NCI designated comprehensive cancer center. And that means we bring a wealth of experience when it comes to not only our breadth of research, but the depth of our research. We're among the country's innovators when it comes to help treating cancer and helping people survive cancer, right? And there are only about 57 NCI designated comprehensive cancer centers in the whole country, and only 2 in the state of Michigan. And we have one right here in our backyard, Karmanos Cancer Institute. But we're not only here in Detroit, we're actually across the state of Michigan as part of McLaren Healthcare. So again, thank you all for being here. I help to oversee our community outreach and engagement efforts and our Office of Community Outreach and Engagement, and this is part of what we do. We say that our job is to connect our communities to our science and our science to our communities. We are so grateful. So we see so many familiar faces here in the audience. Thank you all for showing out. I know that we have a number of people online. How many people do we have online? 50 people online, right, from across the state, so it's wonderful. This is our first time in our new partnership. We are so thrilled to be here at the Detroit Public Library. Thank you so much to the Detroit Public Library for partnering with us and collaborating with us. We are committed to making sure not only that our communities across the state understand our cancer science, but we do so in language that people can understand, down-to-earth, plain language, right? So, because we want to be partners with our community in this fight against cancer. I also want to thank our other partners here today. Many of them have tables in the back. The Pink Fund and Molly, We Run 313, one of the largest run clubs in the state. The Pink Fund, I should mention that again, which offers financial support and other resources to cancer survivors. Healthier Black Elders Center, which is part of Wayne State University. The National Council of Negro Women Detroit section, I'm a member of that. Corktown Health, Cancer Thrivers Network, Western Wayne Family Health Centers, Michigan Ovarian Cancer Alliance, and the New Day Foundation. Thank you so much for being partners in our work. This is the first of 6 talks, community conversations on cancer, and we are thrilled to have today our director of our cancer center, Dr. Boris Pasche. So before we move into our conversation, I wanted to thank Global LT, our company who's providing our translation services today, again in Spanish and Arabic. We are committed to making sure that our materials and our programs are available in different languages for greater accessibility. In terms of people who are in the Zoom audience, we have a Q&A feature, so if you have questions, if you're on the Zoom watching online, you will have an opportunity and a place to ask those questions, and certainly in the room, if you have questions, you're here to ask those. So I think that I've covered everything we're supposed to cover. Again, just really thrilled to have everybody here today. And at this point, I'm going to turn it over to our moderators. They're going to introduce themselves, but they are longtime partners of our Office of Community Outreach and Engagement and of Karmanos Cancer Institute. Just so thrilled that they are here today to help lead this conversation, and we really want it to be a dialogue. I'm going to bring up today Wilma Stringer, Rosalind Sample, and Bill Winkler. Thank you so much.
Hello,
Hi.
I'm Wilma Stringer. I'm an advocate for cancer patients, survivors, and their caregivers. Currently, I'm enrolled in the Build Your Capacity program. Now, this program, sponsored by the Karmanos Cancer Institute and Wayne State University, in case you can't hear me, teaches the fundamentals of medical research to people in the community. This event is an opportunity for me to learn more about advanced cancer treatment. And I want to thank all of you again for joining us this evening.
Good evening, everyone. I'm Rosalind Sample. I am the Community Outreach Coordinator with Pink Fund. And Pink Fund is a national nonprofit. We provide financial assistance to breast cancer patients in active treatment so they don't have to choose between their life or putting food on the table. My passion is connecting with our community, spreading the word about what we do, how we help, sharing resources. I love to share resources because at the end of the day, we all will need a resource. And I hate to turn anyone away. I have been a part of Karmanos and Wayne State and community outreach for a number of years now with these community conversations on cancer. And I love to learn more about how we can help right here on the ground with our neighbors. And that's what we are doing today, is learning how to navigate through cancer with Dr. Pasche's presentation today. So thank you all for coming. I'm so glad to be a part of it. Just some housekeeping before I pass it on to Bill. The restrooms, if you have to use the restroom, they're located on the first floor. You can go right down the stairs outside of this door all the way down, and the restrooms are located right outside there. And then if you take the elevator, you will take the elevator to the second floor, and then when you get off the elevator, you'll go straight to technology, and the restrooms are there. Thank you again.
You got all that information? Where the bathrooms are? Okay, cool. My name is Bill Winkler, and I have been associated with Karmanos since 2011, when Karmanos saved my daughter's life. And since that time, I have been slowly becoming deeply involved in what Karmanos does in the community with cancer prevention, cancer detection, and cancer cure. And I will hopefully continue to do that, and I want to thank you all to be here. We want to educate as many people as we possibly can. I have the distinct honor of introducing Dr. Boris Pasche. I'm going to have to read this stuff because it's big. Dr. Pasche, MD, PhD, joined the Barbara Ann Karmanos Cancer Institute in August 2023 as the president and CEO, and the principal investigator for the National Cancer Institute's Comprehensive Cancer Center Core Grant, and the chair of the Department of Oncology at Wayne State University School of Medicine. That's us. As an experienced hematologist and medical oncologist, Dr. Pasche's clinical and specialties are in treating gastrointestinal malignancies and hereditary cancer. His research focuses on cancer susceptibility and new therapies, which is what Karmanos is about. In 2023, a medical device Dr. Pasche co-invented, Therabionic P1, and received approval by the FDA to treat advanced hepatocellular carcinoma, the most common type of liver cancer. A native of Switzerland, Dr. Pasche earned his Doctor of Medicine and Philosophy from the Karolinska Institute in Stockholm, Sweden, which is the institute that awards the Nobel Prize in Physiology or Medicine. Page 2. He also earned a Doctor of Medicine from the University of Lucerne in Switzerland, following a postdoctoral fellowship at Brigham and Women's Hospital, Harvard Medical School. Dr. Pasche completed an internship and residency in internal medicine at the New York Hospital at Cornell University Medical Center. He's also completed a hematology and oncology fellowship at Memorial Sloan Kettering Cancer Center and a second postdoctoral fellowship at Harvard Hughes Medical Institute and Sloan Kettering Institute for cancer research. Dr. Pasche has given multiple national and international lectures and his area of expertise. He has also published over 150 peer-reviewed articles and book chapters and editorials, many appearing in high-impact journals. Dr. Pasche, you're up.
Thank you very much for this kind introduction. I think I'll take the mic in my hand. That will be simpler.
So excuse me, Dr. Pasche. Before we dive into today's topic, we have to start with a few quick questions to see what we already know or think we know about today's subject. So don't worry, this is not a test, it's not graded, but when if you came here in person, There's a white sheet of paper that's labeled pre-knowledge questions. There are 3 short questions here. You want to take those out. Upon completion, please tear off this sheet and hand it to one of our COE team members, and they'll be coming around to collect it. For our participants joining via Zoom, a poll will be launching on your screen in just a moment with the same 3 questions. So please take a minute to answer them. Thank you. Do you need a poll? Does anyone need a sheet? You had your hand raised. She needs one right in the back there. Thank you. Thank you all for your participation.
Okay, I just learned something. Intermission.
Thank you, everyone. Okay, great. Thank you, Dr. Pash. You can continue.
Thank you. Thank you for the kind introduction. So I will discuss today the development of a novel technology for the treatment of cancer. Cancer is very dear to my heart. I lost my brother of leukemia when he was 21. I was 23. And at that time, I was a medical student at Karolinska Institute. And I pretty much was decided not to do anything to do with cancer because it affected me so profoundly. So fate has that about 15 years later, I found myself during my first rotation as an intern at New York Hospital Cornell Medical Center, landing on the leukemia floor, where a lot of young people, like my brother, were dying of recurrent leukemia, a disease that is still very difficult to treat, acute myeloid leukemia. Only 25% of patients will survive long term. And I remember experiencing this time as some kind of catharsis. I was very sad. I was reliving very painful moments. But one morning I woke up and I decided this is what I need to do. I need to go into cancer because it really affects me profoundly. And I felt a drive to do something new. And I will now present you what this drive had led to 24 years later. So first of all, I need to disclose my conflict of interest. Conflict of interest means that I may benefit from anything that is discussed with this technology because I'm the co-inventor and I hold proprietary participation in the company. So Therabonic Inc. and Therabonic GmbH are 2 companies that I've founded and that I hold stock in. So about me, you heard from Bill Winkler, a very kind review of my CV. So I'm trained both as a scientist and a physician, and I've training in internal medicine and in hematology and medical oncology. During my time at Sloan Kettering, that's when I decided to go into cancer. And during this fellowship also, I decided to go into gastrointestinal malignancies. Why gastrointestinal malignancies? It was probably serendipitous. It turned out that my scientific interests were aligned with those diseases, and I like the people that I work with in gastrointestinal malignancies. So that's a story of me becoming specialized in this field. Then I moved to Northwestern to become the founder of the cancer genetic program at Northwestern Memorial Hospital. Then spent about 6 years in Alabama, where I was the chief of the Division of Hematology Oncology. And then I was appointed as the director of the Comprehensive Cancer Center at Wake Forest prior to coming here. The radio frequencies that I will talk about today are everywhere. Right now, you are surrounded by radio frequencies. Why? Because you have a cell phone. Your cell phone detects the next tower, and you are constantly communicating with the tower. Google knows where you are. The NSA and everybody else knows where you are, where you go, because your cell phone sends back these frequencies. So you are surrounded by frequencies, and these frequencies, you cannot feel them. We cannot feel radio frequencies unless they are very high intensity and would cause heating of the body. But otherwise, no human body can really sense well. Some patients are sensitive to radio frequency electromagnetic fields. That's a very small sliver of the population, but some people feel disrupted by cell phone. So we cannot say that nobody cannot feel them, but it is extremely rare. The device, Therabonic, that I will talk about has been created using radio frequencies, and I will describe in detail how this works. We did research that shows that these radio frequencies can shrink tumor, and I will show you CT scan. They can delay the progression of cancer and support long-term treatment. Actually, the patient who lived the longest with this technology lived 16 years and a half with thyroid cancer metastatic to the lung, to the bone, to the brain, and to the liver. And he passed away after 16 years and a half of progression of disease, but he was only on this treatment for 16 years. In liver cancer, which I will discuss in a moment, we've had patients living 6 years and a half after progressing on standard therapies. So there is clearly something exciting about this technology, is the ability to control cancer, to shrink cancer, and for a long period of time for some people, and as you will see, essentially without any adverse events. So radio frequency, you see on the slide, the spectrum of radio frequencies. You have the X-rays. These one are frequencies that can cause damage to the DNA. You have the visible light, you have the microwave, and we are in this range. We're in the mobile phone range, which is between 30 hertz and 300 megahertz. So that's where mobile phones are. So if you think about radio frequency, your cell phone is probably the closest it comes to the signal I will be talking about. When we look at radio frequencies which disrupt or displace particles of whatever they move through, we don't feel it, as I mentioned, but radio frequencies have some features. The feature is how often do they move, and that's the frequency, and how deep are they modulated, and it's called the amplitude. So amplitude and frequency are the two features of these frequencies. The faster they go, the higher frequency it would be, the lower frequency it would be. So what we are doing, we are modulating this frequency, which shows 27 megahertz as the carrier frequency. Some of you may know 27 megahertz, that's the citizen band. It's also a frequency that is allowed for medical devices worldwide. So it was convenient to use that. So during our examination, we examined patients that were lying on their back, and we exposed them to these frequencies that were being modulated. So we were using 27 megahertz as what we call the carrier. And this carrier, if it were my voice being the carrier, a modulation of 2 hertz would be ow, ow, ow. That would be where you modulate your signal. So we modulated this signal with very precise devices, and we were measuring the pulse of these patients with cancer. To our surprise, we found that patients with the same type of cancer were always exhibiting changes in pulses at the same frequencies. Initially, we thought must be different for everybody. It was not. So we realized, let's say, patient with breast cancer would always have a change in pulse at a given frequency, and this change in pulse was the same irrespective of the age of the patient, whether they had advanced or early stage disease. So what I'm showing you here is the final 422 frequencies that led to FDA approval. This is really the sum of examination of hundreds of patients with liver cancer that showed that with these 422 frequencies, we were covering pretty much everything you would find in a patient with liver cancer when they progress. So this is an example of an individual being tested for these frequencies. So we would put the spoon in the mouth or use an external antenna, and then we would measure the pulse. And as soon as we detected a change, we would refine this change by getting closer and closer. So we started with 100 hertz steps, then we would go down to 10 hertz, then to 1 hertz, and down to the 10 to the minus 3 hertz. So really the thousands of hertz. What we found is that patients who had cancer who was more advanced and had more tumors responded to more frequencies. We also found that patient who had a stable disease, which means a cancer that did not progress or that was responding to therapy, we found virtually no frequencies. So it became clear over a few years of research that these frequencies were a marker for disease activity, for a cancer that was happy and that was growing. If the cancer was blocked, we would see very few frequencies. So that was the first publication we did on this topic, showing the frequencies, reporting the frequencies we identified in multiple types of tumors. So one thing that I want to highlight is the fact that the tumor-specific frequencies, so frequencies that were only found in one given type of tumor, were the majority. As you can see, it's ranged from 70% to 90% for some tumor, which means that these frequencies were not the same for all cancers. And we had only very few frequencies that had common frequencies among multiple tumors. So that's why we started to think, well, there is maybe a tumor-specific frequency or a tumor-specific frequency signature. So after a few years, what we had found is that the majority of these frequencies, I pointed out, 78% were found only in patients with a given tumor, and 22% were found in more than one or two tumor types. So we developed a device that would allow the delivery of these frequencies. So I shared with you, we used 27 megahertz because this was authorized for medical use. Lucky for us, 27 megahertz is a frequency that has some other benefit, which I'll show you in a second. So what we developed is a device, this portable device, which is exactly what you see on this slide, about this side. And this device is connected to an antenna, and the antenna is placed in the mouth of patient during treatment, like you would lick your ice cream, like that. Except of licking, you hold it in your mouth for 1 hour. So that's the treatment. And this device was designed to be very safe. It is operated by a 3.5 volt battery, so there is really no risk of electrocution. It's recharged with this docking station, so patient place the device on the docking station when they are not undergoing treatment, and you recharge that with a power supply. There is also a chip card, and this chip card allows for activation of the device for 1 month of treatment. So patients, when they receive this treatment, have an effect that goes through the entire body, and that's what we call the body antenna. You'll ask why. Well, it's something I learned after we started these experiments, is that in biological systems, such as human body, the best absorption of radio frequencies is that 1/4 of the wavelength. And the wavelength of 27 megahertz is 11 meter. And 1/4 of the wavelength is very, very close to the size of the human body. In other words, when you put this spoon in your mouth, the entire body absorbs these frequencies. That's why it allows us to have a systemic therapy that will touch the tumor wherever it is, both the primary tumor and the metastases. And this is what you see here, is that a patient receiving treatment with a tumor in the liver and metastasis in the lung, a very common side. So the effect of these frequencies is on both the primary tumor as well as the metastases. Here you have a CT scan. You probably have seen that some in your life. And the CT scan is quite telling, because that's where we started to realize we're having a profound effect on the tumor of some patients. This patient received one of the standard drug called sorafenib between March and July and had major progression, so the drug did not work, which is not uncommon. Started the treatment on the device, and you may not see that very clearly, but the primary tumor essentially completely disappeared. There was no more primary tumor. There were also lesions in the chest. You can see one here and one here, and impressive, this metastasis completely disappeared also during the treatment. So we had shown in a patient, and we have several cases like that this treatment was very specific for the tumor. It had an effect on the tumor, but not only the primary tumor, but also the metastases wherever they are. We have had effect on brain metastases. We have had effect on bone metastases. So it appears that these frequencies can deliver the signal throughout the body. So that's why it is a systemic therapy. Dr. Jimenez, who is here in the room, has been working hard identifying the mechanism of action. So once we started to see this tumor shrinking, we asked, but how does it work? Because as you heard, our basis for development was patient examination, discovery of frequency, and then administering these frequencies to patients with cancer. We did not have a preconceived concept of how it would work. So what Dr. Jimenez and his team did was to expose tumor cells to these frequencies and expose tumor cells to dummy frequencies or no frequencies. Dummy frequencies would be frequencies that don't have any effect that we think of in cancer. And what he found is that when we did that, not only we could show that the frequencies, the good frequencies were shrinking the tumor, but he found that there was some calcium coming into the cells. Why did we look for calcium? Because calcium was a mediator that several groups throughout the world had studied in the effect of radiofrequency electromagnetic fields. Fast forward, because it's a long series of experiments, he demonstrated that one receptor called the CAV3.2, a calcium receptor, was the receptor that sensed these frequencies and triggered an influx of calcium only in these cancer cells that led to growth arrest. How did he prove that? Well, he used some drugs that block only this calcium channel, and then the effect was gone. He used also experiments to downregulate, which means to remove this receptor, and it stopped. So we are fairly sure that this receptor is the sole bioantenna, the antenna on tumor that sense these frequencies and trigger the effect. Same data were generated by another group in breast cancer showing that it's the same receptor, even though the frequencies are different and even though the mechanism is different. So to do these experiments, Dr. Jimenez and our two research group used both set up in vitro so that we can do experiments in plates. And as you see, these plates would hold the tumor cells and they would be exposed to either the active frequencies or the dummy frequencies. For the mice, we talked to them. They didn't agree to hold a spoon in their mouth, so we had a problem. So we realized that the only way to do this would be to have a system that replicated. I will stop here for the 30-second pause.
My apologies. We have to allow a 30-second pause to allow our interpreters to transition. Interpreters, please transition now. I was trying to catch it earlier and you couldn't see me. Sorry. You can keep going. Thank you.
So for the interpreters, for the mice, and for everybody else, what we discovered was we could replicate this level of exposure in mice using a system that had antennas beaming frequency from different direction. And with this, we could actually deliver exactly the same level of radiofrequency electromagnetic field in mice as a human would receive. So with these two setup, we were able to do a lot of experiments and really unravel the basic mechanism of action. A very important slide here was taken from tumors from mice that had been exposed to these frequencies. Mice were exposed to either the active frequency, liver cancer, randomly chosen, stand for RCF, or sham, no frequency at all. So we looked at several things. First of all, cell proliferation. And we showed, you can see the staining. The darkest stain here means that these cells are happily growing. When you expose them to the liver cancer frequency, they stop growing. That was significant. We also looked at the intestinal epithelium of the mice. Why do we do that? Because, as you know, for some of you who have received treatment with cancer, frequently you get effect of the gastrointestinal tract, either diarrhea or other problem, because the intestinal cells in the guts are renewing themselves quite rapidly. So when you block them with chemotherapy or any treatment that block the growth of cells, you will have impact on the gastrointestinal tract. Well, what we found is while the tumor was shrinking, the cells in the guts of these mice was perfectly fine. And indeed, we never saw symptoms in patients, and we never saw symptoms in mice that had tumor that were shrinking with these frequencies. So that was another element showing us that it was a targeted therapy for the tumor that did not have any effect on the normal cells. So what data do we have in patients that led to this FDA approval? So it's a total of 59 patients that had been treated in 2 separate studies, and we looked at these patients based on their liver function. In liver cancer, the poorer the liver function, the shorter the survival, because it means your liver does not work very well, and liver failure is a frequent cause of death. Patients who have an excellent liver function and liver cancer live much longer because you can treat the tumor without being slowed down by a liver that doesn't work. So here it shows just the survival of these patients, and you can see the patient with Child-Pugh A, who have the best liver function, lived much longer than those with impaired liver function. The FDA asked to see every single piece of data we had from these clinical trials, and told us to do some additional analysis on this data, and it demonstrated that survival of patients who received this treatment was increased by 34%. And in patients who had impaired liver function, it was just as good as the best drugs in those days, but without side effects. So that's a little timeline of this work that took more than 20 years before we receive approval. In 2022, we received what we call a humanitarian use designation. And in 2023, as mentioned before, the device was approved for all patients who have liver cancer, if they have failed first-line or second-line therapy, or if they are intolerant to these therapies. In 2024, CMS, the Center for Medical Services, gave us a new code, recognizing that this is a novel therapy and there are no code for devices or technology. So this new code became active in November of last year, and that's when we started to distribute the device. The first devices were given to patients at Karmanos. We have now patients in the state of Washington that are also under treatment, and we're expanding nationwide. So Therabonic has also evidence of clinical response in additional tumor types. You may remember this table I showed initially showing breast, showing glioblastoma, showing lung. So these are the type of tumors in which we have had patients experiencing tumor shrinkage while receiving the treatment with Therabonic, programmed with tumor-specific frequencies. So as I just mentioned, we have now patients treated at Karmanos on a standard of care with this treatment for liver cancer, but additionally, we have some very promising data in pancreatic cancer. In pancreatic cancer, we added this treatment to the standard chemotherapy for patients with advanced pancreatic cancer, which means patient with stage 4 disease that has spread already to another organ. And we have 2 trials, one in North Carolina, one at Karmanos, and the preliminary data suggest an improvement in survival that is comparable to what we have seen in hepatocellular carcinoma in the order of 30%. So this trial is open, and if any patient has been recently diagnosed with pancreatic cancer, that's certainly an option that would be available. For any patient with liver cancer, it is available at all Karmanos sites and also at non-Karmanos sites since it's FDA approved. And we have 3 studies that are in progress in breast cancer. We have 1 study in women with breast cancer that were just diagnosed before they get there, between the time of the biopsy and the time of the surgery, so they can receive treatment for 2 weeks. We have also a study for women with HER2-positive disease that are refractory to treatment, and also a protocol for patients with triple-negative breast cancer. These 3 protocols will be activated in the next few months. We have also another trial in colon cancer that is being prepared. And since there is strong activity in prostate cancer, that's another disease we are planning to study in the next few months to a few years. So here I want to thank all the people that did a lot of work in this project, and first and foremost, Alexandre Barbeau, who is the co-inventor of the technology. Together, we examined a lot of patients, identified novel ways to detect these tumor-specific frequencies, developed two companies, one in Germany that builds the device, one in the US that receive FDA approval. And these are the people working for Therabonic Inc. and GmbH. At Karmanos, Dr. Shields is overseeing the pancreatic cancer study. Dr. Kim is the biostatistician, and the other doctors that treat gastrointestinal malignancy, Dr. Saif and Al-Halak, are involved. Dr. Asmi is an expert in pancreatic cancer as a PhD, and we are doing several work together. Dr. Jimenez, whom you see under the Pasche Lab, is now an independent faculty here at Wayne State. He was a postdoc and then an instructor at Wake Forest, and now runs his independent group in researching the mechanism of action this tumor. He just received a new award from the Fibrolamellar Foundation, which is a rare tumor type from the liver that you see in children and young adults, for which we have really limited therapeutic effect. And he demonstrated already in cell line that these frequencies block the growth of these cells. So he's working on that. And I want to thank you for your attention, and I'll be happy to take any questions you might have. So the question was, do you have any plan to study blood cancer? And that would be leukemia. As I told you, that was really the reason why I went into cancer research, and that would be my dream to be able to do that. We have already frequencies for lymphomas, for some leukemias, but we have not examined enough patients, I believe, as of today, to have a robust list of frequencies for these tumors. But I don't see any contraindication. It could be easily done. It does not cause any side effects, so that's certainly something that we're planning to do. Because I'm a gastrointestinal oncologist, and because we have started in this tumor, historically that's where we went, and because we had seen a lot of patients with this tumor, but I believe that there is certainly an opportunity to expand into these diseases.
Okay. Could I just jump right in? You notice that you have a highlighter yellow sheet? Forget that.
My question is, are the radio frequencies from cell phones harmful to us in any way?
It's a good question. As you know, there have been multiple studies on radio frequencies. The overwhelming consensus is that these radio frequencies do not cause DNA damage, do not cause cancer. There are a few studies that have shown potentially an association with neurinoma, which are tumors from the ear canal in patients that use their cell phone a lot, but these associations are pretty sketchy and weak. In general, there was a lot of concern about cell phone increasing the frequency of brain tumors. And when you look at the history, brain tumors started to increase in frequency before the use of cell phone and continued through the use of cell phone. So I would say the jury is still out. I would not say that we have conclusive data showing that it's safe, but the overwhelming evidence is that if it has an effect, it's a very weak one.
Thank you so much for this talk, Dr. Pash. This is amazing technology. My question is, if you have, I'm assuming there's quite a bit of cell die-off with this as you are targeting these cancer types. Do patients experience any kind of a Herxheimer reaction with this?
So very interesting question, and I didn't want to go into these details, but I have good slides showing that, and it's published in the Jimenez et al. Paper that is publicly available on eBiomedicine in 2019. What we did, to answer your question, when we saw this tumor shrinking, and that was after I gave a presentation at UNC Chapel Hill, an expert in liver cancer came to me and said, why don't you tag your cells? So that's what we did. We tagged the cells with a marker called GFP, green fluorescent protein, and we were able to follow the cells. What we found is that the tumor cells transform into scar tissue. Spindle cells morphology, and these cells are essentially eliminated. So we don't see dying off. That was a good question. That was the first test we did. Dr. Jimenez did multiple tests to look for apoptosis, for cell death. We did not see that, but we saw transformation of hepatocellular carcinoma cells into scar tissue and then disappearance of the scar tissue.
Dr. Pash, we have a question from an audience member online. They said, were esophageal tumors looked at in your original work? Can this device be applied to esophageal tumors?
I would say unfortunately not. We have not treated any single patient, and you saw I didn't put it in the list. It's certainly like leukemia, an area we would like to look into in the future, but it is not among the tumor type for which I have data to support an activity.
How you doing, Dr. Pash? I'm thinking of preventative measures. Like, what are some of the things that we can do as a community so that we don't get these type of cancers?
Good question. For liver cancer, one thing is clear: obesity, heavy use of alcohol, smoking, all are risk factors. Chronic hepatitis B and chronic hepatitis C are risk factors as well. Anything that irritates the liver, increases inflammation, increases the risk of liver cancer. So you have, for example, patient, usually it's a disease of elderly people, but we see that children that contracted hepatitis B from a mother that was hepatitis B positive, unbeknown to them, may have had an exposure to this hepatitis B for many years and may develop liver cancer already in their 20s, whereas normally somebody who gets infected as an adult would develop this tumor later. For other type of tumors, I think there is an increase in the frequency of colon cancer. We don't know really why. We know that younger people seem to have an increased risk of several types of cancer. It could be environment, it could be many things, but I don't think we have an answer. And whether this could be used for prevention, potentially, but this is really far down the road. What we do in oncology is to develop technologies for hard-to-treat tumors, see if they work, and then we move up to earlier stages of tumor, and that's what we are doing now. Did that answer your question?
Dr. Pash, we have an online question. The question is, what prompted you to first try radiofrequencies on tumors?
I wish I could give you a 30-second answer, but I'll give you a 2-minute answer. With my colleague Alexandre Barbeau, we started to use radiofrequencies back in the early 90s for the treatment of insomnia. To make a long story short, we had developed this little device and we had found 4 frequencies that worked well on insomnia. So well that we did a randomized study that demonstrated a benefit in patients who had chronic insomnia. The problem was not the FDA. The FDA said, oh, very interesting, you've demonstrated activity. The problem was Johnny Cochran and the O.J. Simpson trial. Johnny Cochran developed brain tumor and everybody was seeing using a cell phone. And suddenly everybody was very concerned about the long-term side effect of radiofrequency electromagnetic fields. So what I didn't share with you is that by 2000, when I woke up one day with the idea of doing that, I was frustrated. I was angry because this beautiful project we have developed was dead because people had concern about the toxicity of cell phone. And one morning, while I was at Sloan Kettering, I woke up and I say, all the drugs we use are so far more toxic than a cell phone. Could we consider using that in oncology? And that's what triggered this. That's the truth.
Yeah. I have a question from the Zoom. Are there cancers that may not respond to your device?
Absolutely,
For
absolutely.
instance, bone cancer, brain cancer, et cetera, sarcoma, anything like that?
So I would say I cannot tell you that one type of cancer does not respond. I can tell you that even with the best frequencies, at best, we get 50 to 60% of patients responding and 40% of patients not responding. That's the way we have developed our technology. We re-examined this patient when they progressed, found new frequencies. And for the liver cancer, for example, that's really the results of examination of patient in progression, but also in progression after using the device, we found additional frequencies. So I would say it's a work in progress. I do not believe it's the panacea. That would be an unreasonable expectation. However, I want to remind you what I said at the very beginning of the talk. We had a patient alive for 16 years and a half, sucking the lollipop every day for 3 hours for 16 years without any side effect, and it contained his disease for 16 years, which otherwise would have killed him within a year. For liver cancer, where when it comes back and you don't have anything, it's usually a life expectancy of less than 6 months. We had a French gentleman that was in our first study that lived 6 years and a half and did not die of progression of disease. His disease was still controlled. He had developed renal failure, and at the age of 86 decided, I don't want dialysis, let me die. And that was his choice, but it was definitely not the progression of disease. So we do have evidence, but I would say small evidence, that we can control the disease in certain patients for many, many years. And these two are the longest survivors.
Can I cut in out of turn? Question. With your 86-year-old patient, was there any connection with your treatment and the renal failure, or was that just
Nope.
one of those things?
He had an infection that was poorly treated and developed renal failure because of a urosepsis that was picked up too late. Yep.
Thank you.
Yep.
With the radio frequencies affecting the calcium channels, does the patient's serum calcium levels have any correlation with how effective the treatment is?
No, because it's really minute amounts of calcium, and it's only from the extracellular to the intracellular, probably pulsed. We can measure it with calcium fluorophore, but we have never seen any change in the calcium level and do not expect it.
Okay. We've heard a lot of information, and if you look, you should have, this time, we can do it, the yellow highlighter form. It's our post-knowledge questions. The first question is, people can feel radio frequency. Is that true, false, or you're not sure? The second question, tumor-specific radio frequencies were discovered through trial and error experiments. True, false, or not sure? For which cancer is the Bionic device approved by the FDA for treatment, now being offered at the Karmanos Cancer Institute? Breast, prostate, liver, colon. Answer the questions, and some of my colleagues will collect.
Does everybody answer the question? Does everybody have it? Yeah. So just to review, just yell it out. Can people feel radio frequencies? No, false. Can't feel them, invisible, you can't feel them, correct? All right. Number two, tumor-specific radio frequencies were discovered through trial and error experiments. True or false? True. It's true. The experiments, that were shown. And then for which cancer is the device approved for FDA for treatment being offered at Karmanos Cancer Institute? Breast, prostate, liver, or colon? Liver.
liver is indeed the only FDA-approved indications that any patient could. The other one are clinical trial. I'm sorry for misleading you.
It's a test for who's paying attention. That's all that was.
No worries, there are no grades. It's all good. It's all good. That's why we talk about the answers at the end, so everybody's clear. Okay? So, are we? Okay. Okay.
No, I think these are a good question, and the answer was indeed, you cannot feel radio frequencies. I highlighted initially that some people, and I've seen them, can feel a cell phone close to them and feel uncomfortable. So it looks like a very small group of individuals may be sensitive to radio frequencies. Trial and error experiments, it's a tough one. It was focused on patients with cancer and identifying specific frequencies. It could be a yes and no, but I would say it's a little bit of both. For which cancer the therapeutic approved, as you know, it's only the liver cancer for now.
So we're at the end of our time. Sorry about that. We're at the end of our time. Thank you, Dr. Pasche, for being here today. Can we give him another round of applause?
Thank you very much for this opportunity.
I want to thank our moderators, Wilma Stringer, Rosalind Sample, and Bill Winkler. And I really want to thank our team here. I just want to acknowledge our COE team. They work really hard every day to make things like this happen. This exceeded our expectations. So thank you for being here. I want to thank Foncille Brown-Miller, Zach Sichon,
Yes.
Candace, Ali Faki, Brittany Dow, Noor Najem, and Maida Herrera, who I know worked really hard to make sure that everything went today, and Madison Pavlicek. Thank you, everybody, for coming. Zach is going to give some evaluations, so please fill out an evaluation before you leave. And also, we'll probably email you to bug you about the evaluations in case you haven't done it. So thank you so much. Please give us your feedback so we can know what to do differently next time. Tell us what you want to hear about next time. Our next one is going to be when? Do you have the flyer? June 25th, where we're going to be talking about the road ahead, understanding lung cancer diagnosis and exploring treatment. So please join us again here. Thank you, everybody.
Good job.